Hi, my name is Travis Yates, and I'm a field application scientist with Personalis. Today, I'd like to share with you how our comprehensive ImmunoID Next platform can enable biomarker discovery, not only for solid tumors, but can also be a solution for heme malignancies. Now, heme cancers are bucketed into three broad categories: leukemia, lymphoma, and multiple myeloma. Unlike leukemia, where sample availability is not often a limiting factor, both lymphoma and multiple myeloma face challenges due to encountering limited sample material. Lymphoma is defined as a malignancy of the lymphoid tissue, and these cancers tend to aggregate and form masses similar to many solid tumors. Multiple myeloma is a cancer of plasma cells, and these malignancies tend to accumulate in the bone marrow. Now, taken together, lymphoma and multiple myeloma are very prevalent heme cancers, and they account for approximately 65% of all newly diagnosed blood cancer cases every year. Now, many conventional next-gen sequencing assays lack the technical capability to process difficult sample types like FFP or fine needle aspirates. Additionally, a comprehensive genomic profiling solution is needed for these cancers, and that's due to the relatively high genetic complexity that they present with. The spectrum of mutations observed in these malignancies is highly variable, and it can range from translocations to insertions and deletions, fusions, and copy number alterations. Ultimately, an effective biomarker solution would require that it is able to profile all of these various alterations simultaneously. Now, at Personalis, we've recognized that there are many issues plaguing biomarker advancement. The first and possibly most important is how to best utilize the limited and precious tumor specimens. Now, to realize a sample's full potential, a multitude of different specialized testing needs to occur. And this is often accomplished using multiple vendors, and it adds to complications in a number of factors, such as time of analysis or difficulties with data integration. Now, taking these factors into consideration, at Personalis, we've really worked to create a more efficient platform and process that keeps sample sparsity in mind while providing a multitude of high-quality data. Our process starts with the initial receipt of limited sample volume, followed by dual extraction of DNA and RNA, and then processing onto our platform to produce a rich array of analytics. ImmunoID Next can be thought of as a one-stop solution that enables multiple analytics from a single sample preparation. Unlike traditional exome and transcriptome assays, the design of ImmunoID Next includes unique features. In addition to the augmentation of all 20,000 genes using our proprietary ACE technology, we sequence at an increased depth for the exome and the transcriptome. We've also added a unique boost to actionable cancer-related genes at 1000x coverage. We've boosted coverage of IO regions such as the TCR and BCR genes from the transcriptome. We've improved coverage of HLA genes. We've placed probes in microsatellite regions to detect instability. We target 7 of the most common oncoviruses and can delineate based on their genotype. And we've worked hard to create a comprehensive assay for immunogenomics profiling. With ImmunoID Next, researchers working with limited samples, in the case of lymphoma and multiple myeloma, will have the opportunity to utilize an expansive set of biomarkers to advance therapeutic development. For lymphomas and multiple myelomas, clinically actionable alterations are consistently observed in multiple pathways. Now, this can range from the PI3K AKT pathway in lymphomas, or the MAPK and Notch pathway in multiple myelomas. Now, for lymphomas, studies have demonstrated the ability to delineate patients' response to BTK inhibitors. MYD88 mutations, which are common genetic abnormalities in some subsets of lymphoma, have been implicated in determining the response to BTK inhibitors. Now, this makes it an important gene to profile to facilitate disease stratification and therapy selection. Now, mutations in DNA damage response genes like P53, ATM, and ATR are common, along with alterations to a number of cell cycle regulators. All of these genes are covered at 1000x to provide enhanced sensitivity and to identify mutations with ImmunoID Next. With the approval of PD-1 inhibitors for a subset of lymphomas and combination clinical trials of PD-1 and BTK inhibitors in the works, really understanding the TCR repertoire continues to be an important marker for blood-based cancers. Gene and transcript and variant level expression profiling remains an important foundation. It's been said that accurate determination of differential transcript usage in genes with many isoforms requires high coverage and sufficient sample. Now that makes ImmunoID NeXT an ideal platform as it provides both deep coverage from limited sample volume. Now both lymphomas and multiple myelomas have characteristic copy number alteration changes, which now these facilitate therapeutic stratification with patients. In the case of lymphomas, BCL2 implications are recurrent. For multiple myelomas, P53 deletions are well known, along with a number of other characteristic deletions and amplifications that have significant prognostic and predictive value in disease progression and clinical outcome. Finally, HLA loss of heterozygosity is gaining importance as a predictive biomarker. With the approval of PD-1, PD-L1 inhibitors for a subset of lymphomas, it would benefit researchers to understand this as a possible mechanism of resistance. Altogether, the oncology field has long suggested the need for next-gen sequencing solutions to aid in identifying genetic alterations in cancer. This continues to become a major area of clinical development and is pertinent when discussing heme malignancies. Now, I hope I've piqued your interest in utilizing ImmunoID NeXT for heme applications. Please check out our newly published brochure titled Biomarker Discovery for Lymphoma and Multiple Myelomas that can be found on our website. And also, if there are any questions, please reach out to us at info@personalis.com. Thank you.